Bismillahirrahmanirrahim. In this lecture, we will discuss about another division of algae, and this is Rhodophyta or Rhodophyceae that are commonly known as red algae. So, in this division Rhodophyta, we will discuss the following contents that are journal account, occurrence of division Rhodophyta, few important examples, a cell structure of Rhodophyta, reproduction of Rhodophyta, economic importance of Rhodophyta, and the classification of Rhodophyta. So, first we discuss some journal account. As the name tells us, they are the red algae. So they are red due to the presence of certain predominant red pigments. The pigments are chlorophyll A, chlorophyll D. We know that chlorophyll A is universally present in all the green plants. So along with chlorophyll A and chlorophyll D, there are R-phycoerythrin. So this R-phycoerythrin is the red pigment which is found in their body and due to which the color of this alga is red. Red algae, as they are large and diverse group of algae, so they contain about 400 genera and 4,000 species. The red algae, they are predominantly marine algae and distributed throughout the ocean. As far as the thallus is concerned, thallus of red algae, it is mostly multicellular. Some of them have complex body organization. The group is considered as one class, the Rhodophyceae, with two subclasses, Benzioideae and Floridae. Now, the occurrence of Rhodophyta. As it is an aquatic alga, so it is normally found in all oceans. Generally, they are found on upper surface of the water, where there is an abundance of light, and also in the depth of the ocean, where there is a little light to penetrate. So they are found in abundance on surface as well as in depths of oceans. They may be found at the depth of 120 meters, particularly in the coral reef areas. And what are the coral reefs? Coral reefs are the ridge of rocks in sea. They are formed by the growth and deposit of calcium carbonate excrete to form a hard skeleton. Now, it is not always that the red alga is red. The color of the alga changes depending upon its location in water. As it is an aquatic alga and it is normally found in all oceans, so when we talk about the color, if the red algae is found in deep water, then the color is red. That means it has more R-phycoerythrin, due to which the alga appears red. Now, what is the reason for this? We know that there are pigments which have the capacity to absorb particular wavelength. So this red pigment, which is R-phycoerythrin, it absorbs the blue-green wavelength, and this blue-green wavelength, it penetrates into deeper water. So if the alga is in deeper water, this is the wavelength which is going to be available for it. And so pigments which absorb this wavelength is red. So in deeper waters, the red algae is going to be red. In shallow water, the red algae is going to appear green, as this is because of more chlorophyll, because of which it appears green. Now, why it is green? When it is in shallower water, because it is going to absorb the red wavelength, which is absorbed by the green pigments, that is chlorophyll. So the color of the red alga changes depending upon the depth in water. So color is variable. This we have discussed and reason for it also. Now this phenomena of change of color is known as Gaidukov phenomena. In this phenomena, the pigment composition changes to increase its efficiency for the maximum adoption of light. So we can say that red algae, they show Gaidukov phenomena. So some red algae, they are terrestrial, forming reddish scums. Many of them are freshwater. Freshwater species are confined to the cold, fast-running water. So red algae, they are most widely distributed geographically and vertically of all the large algae. Most of the red algae, they are autotrophic. Some are epiphytes, that is grows on the surface of other plant, or endophytes, that is which lives inside other plants. And those that are epiphytes or endophytes, some of these may be very pale in color or completely white, and it is presumed that they are in part, if not wholly, dependent on the host for their nutrition. Now, let us come to few important examples of Rhodophyta. The members of red algae, they are the most showy plants. Showy means that they have lace-like appearance and look very beautiful. Here are few common examples of Rhodophyta. You can see in figure Glaceraria and Gelidium. These are the red algae which are commercially used to obtain agar, which is used to make nutritive material solid. Another example here is Porphyridium. It is terrestrial or land algae, forming reddish scum on damp surfaces in greenhouses or the margin of drying garden pools. Another example here is Chondrus or Irish moss. It is edible and it is used as food. The reason why we called it Irish moss, moss is undifferentiated body, and because it was used during Irish famine as food, it is called Irish moss. Another example here is Porphyra. It is also an edible species of red algae. Now, the Batrachospermum. It is a freshwater or river water genus of alga. We will discuss this genus in detail as an example genera of Rhodophyta later on. Last one is Polysiphonia. They are present in quiet water as dense tufts mass, mostly in the littoral zones in tidal marshes. Most of its species are epiphytes on other large marine algae. Now we will come to the cell structure. Because it is a plant and plant has to have a cell wall. Only thing is that instead of pure cellulosic material, there are some other materials that are deposited in it. So the cell wall of red algae, they have multiple things. It has a cellulose and it has a pectic material and some sulfates also. So cell wall is not purely cellulosic. It has other substances along with cellulose. A number of complex colloidal substances such as agar and carrageenan may also occur in the cell wall and intercellular spaces of many species of Rhodophyta. So these phycocolloids, they prevent the alga from desiccation and act as shock absorber against waves. These compounds are mixture of sulfated polysaccharides. In certain genera, there is heavy calcification of outer part of cell wall, which is chiefly composed of calcium carbonate or may also contain magnesium carbonates. In some coralline genera, the whole thallus is encrusted with lime. Now, chloroplast. Central, that is axial, or peripheral, that is parietal, chloroplasts are also present. In some simple forms, only one chloroplast is present with or without perinoid-like structures. Complex forms have small discoid chloroplast in each cell. Now the pigments present in red algae. As we discussed earlier, the red algae have their characteristic red color, and this is because of the presence of accessory phycobilins pigments, and they are generally the R-phycoerythrin, which is of red color, along with that of R-phycocyanin, which is of blue color. So red algae, they contain B-phycoerythrin, phycocyanin and C-phycoerythrin also. Accessory carotenoid pigments are also present. These include two carotenes, that is alpha carotene and beta carotene, and the xanthophyll pigments like lutein, zeaxanthin and possibly teraxanthin is also present. And as we discussed earlier, that the green pigments include chlorophyll A and chlorophyll D, which are generally masked by the accessory pigments. Now nucleus. The cell in simple forms are usually uninuclear, that is, they have a single nucleus. In some advanced form, they are generally multinucleate, except for young or reproductive cells. In some example, large cells of red algae, they contain hundreds of nuclei. These nuclei have well-defined nuclear membrane and a nucleolus. Now, reserve food materials. The stored food is in the form of polysaccharide, that is, Floridian starch. This Floridian starch or a storage product is a carbohydrate which resembles or it is very much similar in structure to amylopectin, which is a component of a starch, and the glycogen. Floridian starch is composed of alpha-1,4 and alpha-1,6 plus alpha-1,3 glycosidic linkage. Other reserved substances of low molecular weight includes saccharides like trehalose, fluoridose, monoglyseric acid, and sucrose. Certain polyhydroxy alcohols are abundant in some forms. Fats are also stored. They include cholesterol, cytosterol, and fucosterol. Now, pit connections are also present in cell structure of Rhodophyta. Cells of siphon are found interconnected by pit connections. Pit connections are the thin portions in the cell wall that adjacent cells can communicate or exchange fluid. So pit connections are the characteristic to red algae. Pit connections may be primary pit connections that occurs between central and pericentral siphons. Secondary pit connections may establish between overlying pericentrals. You can see a primary and secondary pit connections in the diagram shown on the slide. Now, reproduction. Reproduction in Rhodophyta is again by three methods. One is vegetative, another is asexual reproduction, and third one is sexual reproduction. As far as vegetative reproduction is concerned, vegetative reproduction is uncommon in red algae. Except few unicellular forms, it takes place by simple fragmentation. That means that the main body or the main plant breaks into parts or pieces and each part is capable of giving rise to new plant. Now, the asexual reproduction. Asexual reproduction is done by formation of non-motile spores. Various kinds of asexual spores are formed during asexual reproduction. In case of subclass Benguidae, monospores and neutral spores are formed, and in case of subclass Floridae, carpospores, bispores, tetraspores and polyspores are present. As far as sexual reproduction is concerned, it is advanced oogamous type of reproduction. It occurs by the formation of non-motile gametes. Oogamous type means that the egg is stationary or static. Male gamete is non-motile but is carried to the female gamete by water and the female gamete is larger. So this is how this sexual reproduction is take place. There is complex post-fertilization events occur in case of red algae. The male sex organ is called spermatangia or antheridia, which produces male gametes, spermatia. The female sex organs are called carpogonia. Carpogonium is flask-shaped differentiated into swollen base containing egg cells with a neck-like elongated protuberance, the trichogyne. Spermatium carried by water come in contact with trichogyne, where a passage is formed for the entrance of male nucleus. The male nucleus travels down and fuses with female nucleus of carpogonium to form zygote. The act of fertilization is remarkably uniform in Rhodophyceae. One important thing which we have noticed here in red algae that their whole life cycle is non-motile. That is, their spores and gametes both are non-motile. It means in case of red algae there is no flagellated structures. Normally, the spores and male gametes, they have flagella for moving from one place to another. But in the case of red algae, gametes as well as spores, they are non-motile, so there is no flagellated structures. Now, let us come to the economic importance of red algae. Coralline algae like Corallina, they are important in building coral reefs. The reproductive cells of the red algae are liberated in great number and form a part of the phytoplankton. For example, single tetrasporophyte of some species may produce about 10 crore tetraspores. Red algae like Rhodominia, they produce 1 crore 20 lakh carpospores. The red algae, they provide directly or indirectly as a source of food. Porphyra is edible and used as food. They are used as food in Japan. In Japan, it is known as nori and is prepared as dry flavored products. It is also used in preparation of Japanese macaronis, in soups and sauces, or in vegetable as a flavoring agent. So there are certain substances which we obtain from red algae on commercial level and we keep using for various purposes. That is funier, which is used in preparation of water-based paints, as in adhesive in hairdressing and as starch in laundering. Another important material is agar-agar or agar, which we normally use are obtained from red algae, that is Giletium and Gracilaria. On a commercial scale, it is widely used in microbiology in the preparation of media for cultural studies. It is used as therapeutic agent in intestinal disorder as it is non-irritant bulk producer that can absorb and hold water and at the same time act as mild laxative. It is also used to make capsules to enclose antibiotics, sulfa compounds, vitamins and other compounds where a slow release of medicine is required beyond the stomach. It is also used in dental materials. It is also used in certain special breads for diabetics. Agar replaces starch. However, agar is not good for human consumption because it is relatively indigestible. Last material that is used on commercial and industrial level is carrageenan, one more substance which is extracted from all the red algae and it is actually a hydrocolloid. It is a mucilaginous extract used in the food industry as stabilizing or thickening agents in chocolate milk, in cheese, ice creams and jellied food. It is also used in cosmetics, insect spray and water-based paints. Now the classification. Fitch in 1945 divided the class Rhodophyceae into two subclasses, that is Bangioideae and Floridae, and 7 orders. Subclass Bangioideae has one order, that is Bangiales, and subclass Floridae has 6 orders, that is Nima, Leonales, Gellidiales, Cryptonemiales, Gycatinales, Rhodymeniales, and Ceramiales. Important examples of red algae, as we discussed earlier, are also mentioned here. So class Bangiales and Nemalineales, they have haplobiontic life cycle. That is the life cycle in which diploid phase is followed by two dissimilar haploid generations. For example, the gametophyte haploid plant produces the zygote during sexual fusion. The zygote undergo meiosis and produces haploid carposporophyte, which is parasitic on gametophyte. The carposporophyte produces haploid carpospores which germinates to form haploid gametophyte. So here only one diploid generation which is zygote, while the haploid generation is independent gametophyte and parasitic carposporophyte. This life cycle is haplobiontic type. So, in subclass Floridae, Jellydiales, Cryptonemiales, Gigartinales, Rhodymeniales and Ceramiales, they have diplobiontic life cycle, that is the life cycles in which the haploid phase is followed by two successive diploid generations. For example, the haploid plant, that is gametophyte, produces diploid zygote after gametic fusion. This zygote produces diploid carposporophyte on the gametophytic plant. That's all about the division Rhodophyta. Next we will discuss about Batrachospermum as an example genera of Rhodophyta.